Okay, so good evening and welcome to today's class. We will be beginning week 14 or class 14 for this course on genes, genomes, and the epigenome. Let me put this into presentation mode. So, till the last class, what we have been discussing has been different sorts of topics where we have already covered gene structure, function, and some part of gene regulation. The rest of it I will be discussing in today's context. And then we moved on to mutations, polymorphisms, and functional genomics, introduction to sequencing methods, Sanger versus NGS. Until this part, we have already covered this, not just the text, but also have had a examination regarding the same in the past session on Tuesday. Next, we moved on to unraveling the genome, exome, and transcriptome. We understood the way in which the chromatin is packaged, how does it work, what are the different ways in which what are the different tools which you can use for unraveling the genome, exome, and transcriptome, what are the key steps that are part of it. We discussed pre-processing of sequencing data. We first understood how sequencing strategies work, what are the different attributes you should know at that point of time. Then we understood how you need to optimize them based on what your objective is, what sort of hypothesis you have designed. And later we moved on to identifying, um, uh, we moved on to the steps which are necessary for identifying any contaminants, any, external sources which have come in, any um, microbiome associated DNA information or RNA information, all of that we understood. Then we understood how from that raw read you can trace it back to the location from which it possibly has been derived, which is otherwise known as alignment of reads and annotating them to their original location within the genome. Then we moved on to introduction to epigenetics. We understood we had some discussions on DNA methylation and histone modifications, and we eventually will move on to mapping DNA methylation in the next couple of classes. But for now, we will first finish our molecular biology part and we'll discuss decoding DNA protein interactions. And before we move on to the techniques which are commonly known as high throughput, we'll begin our discussion on DNA protein interactions, the necessity of them, some molecular insights into them, how they are so important in the normal physiology of the cell, and what needs to be understood regarding that before we can proceed with understanding high throughput sequencing data and its analysis. So, with this introduction in mind, we will move forward. So, when we talk about DNA protein interactions, from a molecular point of view, lifting the veil from the way a DNA molecule binds to a protein, and basically think about it in this way. If we get a historical perspective into this whole thing, when people initially started understanding that DNA is so largely controlled and regulated by the protein which binds to it, understanding those interactions and manipulating them to our own studies to address our objectives, that was something that scientists were very interested in doing. So Leng and Felsenfield, these were two scientists who were the first to discover that polylysine polypeptides, which you might think of as very small proteins, but do not have any defined 3D structure, they preferably interact with AT-rich DNA, while polyarginine connects desirably with GC-rich DNA. This was one of the first studies where people started noticing that there are certain patterns and certain types of amino acids from certain types of proteins preferentially bind to some sequences, some common motifs, or some stretches of DNA which are more biased towards uh, certain nucleotides over others. Then a decade later, Seaman et al. They sheds a little more light upon the structure of these interactions. And then, using the hydrogen bonding atoms identified by these researchers on DNA-based edges, they suggested that some specific amino acid side chains they are able to recognize certain nucleotides and that there's an increased likelihood that these interactions are more specific in the DNA major groove than in the minor groove. So, overall, what people understood are that there are that certain regions of the DNA, first of all, before we move too fast into it, we have not really discussed the major and minor groove of the DNA till now. We have just understood it briefly in our very initial discussions on decoding the structure of the B form of DNA, the double helical structure. And we have seen that there are certain regions of the DNA which form bigger grooves and then smaller grooves. And there are certain amino acid side chains which are preferentially able to recognize some nucleotides, and these interactions are found to predominate in the major groove of DNA, and they are the preferred site of interaction with proteins. Nowadays, despite knowing that there are some preferred interactions, it is acceptable that protein families, it's not just one individual protein, but some families of proteins which possibly they share some common motif or something of that sort. So, such proteins they preferentially bind to DNA in different manners depending on what sort of the complex is formed, and it's not always a straightforward correspondence that there's a stretch of DNA and on that directly a protein binds. No, it's not as uh, straightforward as that. There are grooves of DNA and there are caveats within the protein. There are certain domains, there are certain cavities which allow the looping of DNA and which allow the DNA protein interaction to be facilitated at a molecular scale and make that interaction as strong as possible. And then there becomes an increased likelihood that these interactions are necessary physiologically for some purpose, and hence there is such a tight control over them. So, a majority of solely chemical investigations related to DNA protein binding, which were performed relatively more recently, they focused on a specific interaction, and you will find a lot of information regarding that as well. Such as the protein's size, the DNA binding site, the strength of the binding, the effects of the protein binding on the structure of DNA, individual groups or specific bases which are involved in that interaction, all of that will be understood. So we'll get into more and more detail about this in today's class in the upcoming slides. But this image that you see on the right, it shows you it's a very generic image of a DNA protein interaction. You do not have to worry about the orientation and all of these things. We'll understand it very soon in the next couple of slides. But basically, this is a sort of it's a modeled structure of DNA where you can use tools like PyMol or Chimera, etc., which are available. I think they're at least some versions of them are free. So you can use these tools and you can put in your sequence and then you can model it using these tools and you can check how your protein potentially looks like in 3D structure in vivo. Of course, keep in mind that these, no matter how efficient they would be, it is still a prediction, and the actual structure decoding requires different strategies. You have to experimentally validate whatever you are seeing, and that is an it's extremely challenging task that becomes one person's PhD. So we cannot really claim much about it at this moment. But right now, what you can keep in mind is that you can see that there are certain regions of the DNA where the groove is larger, and these are the regions where preferentially these proteins will be able to bind because that facilitates more interactions in that location. And the minor groove has it poses to some extent some steric hindrance to these proteins from binding. It is not a preferred site for docking. Its sequence specificity is not ideal for interactions usually, but of course, there are some exceptions to the rule. What we are saying is the most default or the most generic case. Now, apart from that, if you look at the protein structure, you will see that there are some ribbon-like structures. That's just because of the way in which this protein has been represented. It has been shown as a ribbon diagram, and you can see that there are two different colors. Now, what does that signify? It means these are two chains possibly. Now, what do I mean by chains? If you have if you've read about insulin, the structure of insulin, A chain, B chain, and the C peptide is present. Similarly, there is insulin also, which has four individual monomers, which are together forming a tetramer. So these proteins are often made up of multiple chains which assemble together in the 2D and 3D conformation to give the protein its final functionality. So in this case, the yellow and this blue are encoded by different chains, but eventually they're forming a structure that is capable of binding to DNA for some purpose that it serves downstream. So that is how this structure kind of looks. Now the question is, where do we why are we so concerned about these DNA protein interactions, particularly for one reason, that is because in the cell this actually constitutes a very large percentage um, of regulatory mechanisms in inside a cell, and most commonly it was initially studied and was later elucidated as well that genetic switching occurs within cells, which means that not all genes are constantly expressed at the same rate at the same time within a cell, and there is a control which is kept on it. And this it's sort of like a genetic switch which is able to sense that under X conditions this protein must not be expressed, and under Y it must be. So that fine tuning or that control is is it falls under the purview of this genetic switches. Now the question is, how does a cell determine which of its thousands of genes to transcribe? So where did we come to this question? Come back here to the legend again. So when complex or simple genetic switching devices are found in all cells, and they're composed of two fundamental components. What are those components? First, there should be some short stretches of DNA of defined sequence, and there would be gene regulated proteins that recognize and bind to this DNA. Now, if it has, now why is this transcriptional control or regulation so important? It is important because a cell at any given point of time, considering that it has around 30,000 genes, yeah, um, it needs to prioritize that under which conditions, which set of genes should undergo transcription, or which set of genes should be expressed. So one of the most important concepts is based on a group of proteins known as transcription regulators. What are these? As the name suggests, they are transcriptional regulatory proteins. They recognize specific DNA sequences, usually 5 to 10 nucleotides long, and they're often called as cis regulatory sequences. And typically, they must be on the same chromosome that is in cis to the genes that they actually control. So what do we mean by that in cis? Basically, we mean that on the same chromosome upstream or downstream of your gene sequence, many a times you will have these regulatory elements which would be present, and they're usually part of the same chromosome, or they usually insist with the gene that they are controlling. Now, transcription of each gene is in turn controlled by its own collection of cis regulatory sequences. Typically, these lie very close to the gene, often in intergenic regions directly upstream from the transcription start point site of the gene. So if you remember, let me draw this here. You have, say, suppose any random region of or stretch of DNA, and you have this exon 1 and you have this exon 2, and upstream of exon 1 is the transcriptional start site. Now, just hypothetically speaking, it is possible for regulatory elements of transcription to be present somewhere here upstream or insist to the actual sequence that we are controlling. Many a times it might so happen that there are internal transcription or translational sites, and or there might be multiple transcription start sites as well, then in that case, the regulatory element can even in fact lie in the intergenic region, and sometimes even irrespective, you can find it in the um, intergenic region. Now let me clean this up. Okay. Now, how do these different base pairs in DNA get recognized from their edges without the need to open their helix? So, how is it possible that the double helix does not need to be opened and still different base pairs in DNA are able to recognize from their edges? There are four possible combinations or configurations of base pairs which can be shown with potential hydrogen bond donors indicated in blue, potential hydrogen bond acceptors, mostly represented in red, so on and so forth. So basically, there are different base pairs of DNA that can be recognized from the edges, and that's how you can detect it further. The outside of a double helix. Now comes the fact that what exactly is making this reaction work. Just a second. Yeah, so what exactly is making this reaction work? The outside of interaction work, the outside of the double helix is usually studied with DNA sequence information that the transcriptional regulators are able to recognize. Give me a moment, please. I think I had a different slide on this. Mm, No, I cannot. Something like this. Yeah. So, like you can see, we had already mentioned transcriptional regulators here, which are able to bind to cis regulatory sequence, and they actually form a part of the gene regulatory region or the gene control region for any gene X. And here's what you see, gene X, and yeah, and here's whether the transcriptional initiation complex, which includes the RNA pol 2, the sigma subunit of RNA pol 2, general transcription factors, coactivators, etc. An intricate network of all of these are able to bind and they're able to control the expression of gene X. So when we look at the gene control region, then we see that for a typical eukaryotic gene, upstream of the transcriptional start site, you can find multiple sequences or multiple regulatory sequences to which these transcriptional regulators may bind. Now, keep in mind, it's not always necessary that the binding of these elements is going to be a positive, like a positive or a upregulatory, or let me put it this way, an amplification signal, but instead sometimes it can be a retardation or a silencing or a repressive signal as well. Now that depends upon A, what type of element is able to bind, and B, what is the stimulus which has come into the cell, which will dictate this response. And if you remember, we had also discussed something. Yeah, this is where we saw the same as well. That for the Yeah, there can be sometimes there can be the presence of insulator proteins which might be present, and these insulator proteins can actually loop like sort of block some regions of the genome, thereby controlling how the genome expression may be regulated inside the cell. So, coming back to our session for today, let me just quickly go back there. So, the outside of the DNA double helix is studded with the DNA sequence information to which a transcriptional regulator is able to bind. What is that giving rise to? That is giving rise to a structure where the outside sort of serves like a pipe. The inside is the home of all of the possible interactions between nucleotide bases. The edge of each base pair represents a distinctive pattern of hydrogen bond donors, hydrogen bond acceptors, and hydrophobic patches in both the major and minor grooves. Again, reiterating because the major groove is wider and displays more molecular features than the minor groove. Nearly all transcription factors make most of their contacts with the major groove, as we have already discussed. The binding of a transcriptional regulator to a specific DNA sequence on the oh, there is some. Okay, I think I've accidentally put the legend for a different figure. Let me open up Albert's for you. Everything that I've included today has been taken from Albert's Molecular Biology of the Cell. Yeah, so if you see this image, you will see that a single contact, this is your DNA binding protein, this is your DNA. If you are looking at it head on after sort of chopping or slicing through it, then a single contact is present between this transcription regulator and DNA. And such contacts, they allow the protein to read the DNA sequence. So this is your protein, it is able to read the DNA sequence. How is it able to read it? Because of the presence of the side chain or the substituent group or the side chain, which is a part of the DNA binding protein. It is able to form certain types of interactions only if a certain type of sequence is present uh, inside the inside the groove where it has extended its protrusion. On the right, the complete set of contacts between a transcriptional regulator, who here is a member of the homodomain family, and its cis regulatory sequence is shown. The DNA binding portion of the protein is 60 amino acids long. Although the interactions in the major groove are most important, the protein is seen to contact both the minor groove and the phosphates in the sugar phosphate backbone. So if you see here, this is sort of a zoomed in region over here, and it is forming this sort of interaction via the major groove, not via the minor groove. And yeah, protein can sometimes also contact the minor groove as well as the sugar phosphate backbone of the DNA, which we are studying also. So that is what we saw here. And yeah, so that's what a major takeaway are these things only. Then moving forward, when we talk about DNA binding proteins, what do we mean by this group? So they actually form a class of proteins which can have different attributes. What do we mean by attributes? We mean the sort of secondary structures or the motifs that these proteins are able to form will dictate how they're able to A, access DNA, B, bind to it, and C, perform the function that they're expected to do. Now, the question is why at all do we need or why are there these multiple different sort of forms? The question is because if not, what would happen in that case? If not, if all proteins had to have the same sort of structures, all DNA binding proteins are the same sort of structure, then you would be limited as to the number of amino acids which can occur at a particular site, and the amount of homology between very diverse proteins would increase in that case, which can compromise the integrity of the genome. It can make the genome repetitive, and that would certainly not be desirable considering the amount of energy that the cell has expended in sustaining it. So, therefore, multiple families have come up till now. This is subject to change as well. Uh, so, among the different families, the first one is of helix turn helix proteins. Otherwise, sometimes you might briefly see it written as, sorry, yeah, helix turn helix proteins. So, originally found or identified in the bacterial transcriptional regulators, this motif has since been found in many hundreds of DNA binding proteins from both prokaryotes and from eukaryotes as well. It is constructed from two helices shown here in blue and red, which are connected by a short extended chain of amino acids, which constitutes the term. The two helices are held at a fixed angle primarily through interactions between the two. Yeah, just a second. Yeah. Primarily through interactions between the two helices. So the more C-terminal helix, which has been shown here in red, is called as the recognition helix. Why so? Because it is able to sort of um, yeah, it's able to sort of scan through and detect the capacity of the binding in this case. And the more C-terminal helix is called the recognition helix because it fits into the proper groove of DNA. And yeah, its amino acid side chains, which differ from protein to protein, play also an important role in this recognition of specific DNA sequences to which the protein actually binds. All of the proteins which have been shown here, they can bind DNA as dimers in which the two copies of the recognition helix are separated by exactly one turn of the helix. So again, let's come back here. So what do you see? You see that proteins here that we have already discussed, they bind as dimers. Just a second. Yeah. So so the two helices that we are talking about in a helix turn helix motif or proteins, DNA binding proteins which belong to the HTH family or helix turn helix family, there are two fixed angle proteins, fixed angle helices, of which one serves as the recognition helix, as I already discussed, shown here at the top in red, which sort of moves into the minor groove of DNA and identifies the binding capacity, and then allows recruitment of the complete protein. It plays an important part in recognizing the specific DNA sequence to which the protein binds. Now, all of the proteins shown here are able to bind to DNA as dimers, in which the two copies of the recognition helix are separated by exactly one turn of DNA, and both of them can fit into the major groove of DNA, thereby allowing complete recruitment and more specificity. So if you look at the structure also, you will see that there's this recognition helix and there are two such entities present, helix, turn, helix. Um, there is another one which is HLH also, we'll discuss it later, helix loop helix. And so that's pretty much what you see. There's assembly and there can be binding. And they usually bind as dimers. dimers also. Like we can see, there are two recognition helices present for that purpose, and in which these two copies of the recognition helix are separated by exactly one turn. So you would have one recognition helix binding at one side, the next at one major groove, and the next one binding at the next, so as to maintain the pattern, the patternness or the order in which this binding actually occurs. Now, moving on, the next subtype of DNA binding proteins includes this particular group of yeah, okay, let's continue with this first only. So not long after the first transcriptional regulators were discovered in bacteria, genetic analyses of the fruit fly Drosophila led to the characterization of an important class of genes, the homeotic selector genes that play a critical part in orchestrating fly development. And it was later shown that these genes they coded for transcription regulators that bound DNA through a structural motif named as the homeodomain. In fact, my PhD also is on a homeodomain protein. So basically, what they do is they are able to they possess a particular structural motif, and they belong to this family, which has been studied and been extensively annotated in Drosophila, and there it plays an important role in patterning, in development, neurogenesis, etc. Two different views of the same structure are shown. In A, the protein structure is, yeah, the protein homeodomain structure is folded into three alpha helices, which are tightly packed together by hydrophobic interactions. The part containing helices 2 and 3, this is helix 2 and helix 3 in blue and red respectively, this part very closely resembles the helix turn helix motif. The recognition helix, shown here as helix 3 in red, that forms important contacts with the major groove of the DNA. So if you look at this figure, you would see that yeah, recognition helix 3 is able to form important contacts with our host DNA, and yeah, the asparagine or ASN of helix 3, for example. For helix 3, if you see here, it has this asparagine shown here, ASN. Um, contacts an adenine. Now, a flexible arm attached to helix 1 forms contacts with the nucleotide pairs in the minor groove. So that was the second application of the first one. Next, you have the leucine zipper motif. Now, this is a very interesting name and a very interesting story as well. The leucine zipper motif sort of resembles the zipper-like structure that you often find in your denims, etc. And it is named so because of the way two helices, one from each monomer, are joined together to form a short coiled coil. So, coiled coil basically is a second level coiling of already existing coils. Mostly you would find this in protein tertiary structure, the presence of a coiled coil structure. One example, I think if I'm not wrong, then coiled coil structure is often seen for collagen, where you have three different coils which are all which are all sort of mushed into a master coil, but there is in coiling among each of these coils as well. Now, these proteins they act as dimers where the two long helices are held together by interactions between hydrophobic amino acid side chains that extend from one side of each helix. Now, just beyond the dimerization interface, the two alpha helices can then separate from each other to form this sort of Y-shaped structure, which allows the side chains to interact with DNA at the major groove, and the dimer that sorry, the dimer that grips the double helix is much like a clothespin on a clothesline. So basically, think of it as two tongs which are able to bind to DNA upon recruitment, and they act as transcriptional regulators as well. Next, you have beta sheet recognition motif. So, what have we discussed till now? We discussed helix turn helix. We discussed an application or a sort of subgroup among helix turn helix proteins. So that includes the homeodomain group of proteins. And then you saw the leucine zipper. It's sort of this is the dimerization interface. The two dimerization interface have amino acids which are able to bind with each other and form a stable structure. And beta sheet recognition motifs. In the other DNA binding motifs displayed in this panel, al alpha helices are actually the primary mechanism that's used to recognize some specific DNA sequence. In one large group of transcriptional regulators, however, a two-stranded beta sheet with amino acid side chains extending from the sheet towards the DNA reads the information on the surface of the um, major group. Yeah. Now, as in the case of a recognition alpha helix, much similar to that, this beta shift motif can also recognize many different DNA sequences. The exact one that will get recognized depends on which amino acid sequence is actually forming the beta sheet. Now, if you look at the schematic here, you will see that there's a transcriptional regulator that both um, yeah, that binds to two molecules of SAM, which is a adenosine methionine, and on the left is a dimer of the protein. On the right is a simplified diagram that shows you a two-strand beta sheet bound to the major groove of the DNA. Now, that was, if you look at the difference between them, it is not just the orientation, but it is in fact the sequence which makes up the beta sheet that will make all the difference. So, if you see here, you'll see two molecules of S-adenosyl methionine shown here in red are bound to the DNA binding protein, and the second one it's a dimer of the protein, and on the right is a simplified diagram that shows just the two-stranded beta sheet bound to the major DNA group. Next come the helix loop helix proteins, very close and very similar to the leucine zipper. The helix loop helix motif consists of a short alpha helix once again, but instead of being followed by a turn, it is followed by a very short stretch of sequences known as the loop. Yeah. So it consists of a short alpha helix connected by a loop to a second longer alpha helix. The flexibility of the loop allows one helix to fold back and park against the other, forming the dimerization interface. So if you see here, there is dimerization which is occurring, and these two helix structure they both bind to DNA, and to the two helix structure, yeah. So two alpha helices that extend from the dimerization interface, they make specific contacts with the major groove of the DNA. We'll come to the next class of DNA binding proteins. So the next class and the last class of DNA binding proteins, broadly speaking, are the ZFNs, which are zinc finger proteins. So this group of DNA binding proteins includes one or more zinc platforms or zinc atoms as structural components. This group of DNA binding motifs includes one or more zinc atoms, which must certainly be present. Uh, all such zinc coordinated DNA binding motifs are called as zinc fingers, which refers to their appearance in the early schematic drawings. So, if you think about it, it looks much like a um, sort of a victory sign. So, they have two zinc driven or zinc associated uh, yeah, zinc associated DNA binding motifs are present, and they fall into several distinct structural groups, only one of which we consider here, which has a simple structure. So this is the structure if you see, and it includes a zinc atom at the very center, and it is bound by, or it is stabilized by two two hydrogen bonds, and that gives it the integrity. It has a simple structure in which the zinc atom holds an alpha helix and a beta sheet together. This is often found in clusters with the alpha helix of each finger contacting the major groove of the DNA, forming a nearly continuous stretch of helices along the groove. In this way, strong and specific DNA protein interactions are built up through a repeating basic structural unit. Three such fingers are shown on the right. So if you look here, so if you see here, C and H here are representing, I apologize, it was a mistake. H here represents two histidine molecules. C here is representing two cysteine molecules. So these two cysteine and these two histidines together are stabilizing the zinc molecule, or are allowing the zinc to bind and then stabilizing it by with these interactions. And yeah, so that gives it an architecture which is good for zinc finger proteins. Similar to that, if you look at this protein, you'll see that with respect to the DNA, you'll have these three zinc fingers to be present, and you'll see that at the core of these, you will have a zinc ion, which is definitely present there. Next, we'll come to transcriptional regulators, but I will take a short break and be back.
Okay, so till now we have discussed these following. Just a second. Yeah. So till now we have understood how DNA protein interactions are facilitated, what is the conceptual basis for the same, what are the different types of interactions which stabilize the same. You will see that there are ATGC interactions which are stabilizing the DNA, and on its exterior or on the outside of this phosphodiester backbone is where the protein is actually able to bind. And proteins many a time have certain structural features which aid their ability to facilitate these interactions. We looked at um, some of these structural attributes that includes the presence of certain domains. One of this is the helix turn helix domain or helix turn helix family of proteins, whereby they have these two helices, whereby they have two helices which are separated by a non-specific turn. When we say non-specific, we basically mean that these they do not form any secondary structure, and they sort of form a basic simple loop, so to say. Now, this motif, the presence of this motif is usually found to be quite abundant in large number of proteins. Why so? Because of the nature of the protein binding. What do we mean by the nature? We mean that usually what these proteins do, they usually bind as dimers, and their binding or their 3D sort of orientation is such that these red colored recognition motifs that we talked about are able to bind the DNA only at the major groove, which makes which facilitates the presence of two subsequent recognition motifs at two subsequent major grooves, which completes one turn of DNA. And therefore, the ability of these DNA binding proteins to recognize that DNA motif improves by a lot or increases by a lot. Then, uh, from this major class, we discussed a certain subgroup, and that subgroup has been largely studied in Drosophila, which are known as these homeodomain proteins or homeobox containing proteins. They encode for transcriptional regulators, which are able to bind DNA through structural motifs called as the homeodomain. Now, two different views of the same structure have been shown if you look carefully here, and the interaction is stabilized by the recognition motif binding to uh, certain features of the DNA, majorly the presence of amino acids like serine, arginine, and asparagine on the recognition motif. Now, again, keep in mind that this cylindrical structure is again for simplification. Instead, these helices also can sort of look like this, only their ribbon diagram only, and on the outside projects their substituent group for the amino acid, which is being talked about, and then the amino acid which is present, like serine, the OH group of serine, for arginine, the charged group, asparagine, the charged group, they are all able to form nice stable interactions with the phosphodiester backbone, which carries a negative charge. So that stabilizes such DNA protein interactions. Following that, we discussed the leucine zipper motif because it has, again, yeah, the dimerization interface has multiple leucine residues which are present, which is, sort of attributes the name to this class of proteins. And this includes two alpha helices, one from each monomer, which are joined together to form a short coil structure. And these proteins are able to bind DNA as dimers, where the two long alpha helices are held together by interactions between the hydrophobic amino acid side chains, often present on leucines that extend from one side of each helix, and they separate from each other. They form this sort of Y-shaped structure, like you can see. Like if I sort of draw this here, so in contrast to what you see here, sometimes you might also find their drawing to be shown sort of like this, much like an antibody. So these regions are where you have your leucines present, and as a result, they are sort of able to interact with each other. And this is the DNA binding region. And so it sort of gives this a Y-shaped structure similar to an antibody. So that is what we see here. And we also have beta sheet recognition motifs where instead of an alpha helix, if beta sheets are present many a times, you might find that also to interact with DNA. So what does it do? It allows for these transcriptional regulators carrying the two-stranded beta sheet. Again, someone might debate that two-stranded two beta strands do not necessarily form a sheet, but this is the terminology which is used. These beta sheets are also able to read information on the surface of the major groove. Um, Similar to the recognition alpha helix, like we saw, the beta sheet motif can also recognize many different DNA sequences, and that depends which one it will recognize depends on which amino acids are present on the beta sheet. For example, if you see here, the transcriptional regulator, for example, shown here, is able to bind two molecules of S-adenosyl methionine or SAM, which is a universal methyl donor. On the left is a protein dimer. On the right is a simplified diagram showing just the two-stranded beta sheet bound to the major group of the DNA. And then we talked about helix loop helix proteins. So the major difference between the two is the fact that for a helix loop, the loop is much smaller than a turn. Sorry, the loop is much larger than a turn. A turn was simply, say, four bases. I have not gone into the details here, but if you have read the basics of protein structure from Brandon and Toombs, there you'll be able to see the differences between different types of turns that potentially can be present in a secondary structure motif. So a turn can usually have four amino acids, and depending on which amino acid is present, that turn can also be classified type 1, type 2 turns, etc. However, when a large and relatively longer number of amino acids are present, which are not able to form any stabilizing interactions that allow the formation of an alpha helix or a beta sheet, or any sort of stability is not possible, but instead it sort of acts as a flexible loop, then in a flexible portion of the protein, a polypeptide sequence, then many a times that is referred to as a loop. Now, there are two schools of thought also about this. The presence of a loop sometimes can be considered to be simply an unstructured region of a protein, but over time, more and more studies have shown that they are not just some pointless, uh, it's not an energy wastage for the cell to go through the translation of such loops, but instead their presence many a times is necessary to facilitate very important interactions. The flexibility of this loop makes interactions a lot more dynamic, and their flexibility also allows them to assume different shapes and facilitate different interactions, which might be more difficult for a more rigid structure to do, and therefore their presence is also very important. So it consists of a shorter alpha helix connected by a loop, and then a longer secondary alpha helix. So this is the short one, the presence of the loop, then a long one. This flexibility allows one helix to fold back and part against the other, forming the dimerization surface. Again, the dimerization is much similar to that of a leucine zipper. And as shown, this two helix structure binds both to the DNA and to the two helix structure of a second protein. So you have two independent chains or you have two independent proteins which can together form this dynamic interaction, and it can either be a homodimer or a heterodimer depending upon what sort of protein it is. Now, two alpha helices that extend from the dimerization interface make specific contacts again with the major group of the DNA. So this part remains constant. The site of interaction remains the major group of DNA. Why? The reasons we've already discussed before. It is simply the motif that the context of the motif binding and the regions of the motif that can actually be accessed, that is differing among different classes of proteins. But the overall concept sort of remains the same. Then we finally moved on to DNA binding protein family known as ZFNs or zinc finger proteins. Why ZFNs? Most commonly, their nomenclature is usually in this manner, ZNF or ZFNs, and they include their structural stability and their functionality is very heavily reliant upon the presence of zinc atoms in that structure itself. And what it eventually forms, this chain of polypeptide forms, resembles something of like that of a finger, hence the name zinc finger proteins. And they form coordination bonds. Zinc atoms are able to form coordination bonds with histidine residues and cysteine residues, which allows again a lot more structural stability. And using these motifs, they're able to bind to DNA. And again, if you look here, this is your DNA phosphodiester backbone, and close to it, you have this recognition helix, and you have your zinc atom at the center, which is forming coordination bonds. And often you will find that they fall into different structural groups as well. If the zinc atom holds an alpha helix and a beta sheet together, what you see here, this is your beta sheet, this is your alpha helix, and at the center, there is a zinc atom which is forming coordination bonds and completing the zinc finger. That is one possibility. The other possibility exists that there can be two different helices, and the zinc atom coordinates between those two as well. And yeah, this forms a continuous stretch of alpha helices. And so a strong and a rather specific DNA protein interaction can be built up by repeating such basic structural units. And usually you'll find three zinc fingers which can be present, and they are able to bind to DNA at three subsequent major groups, which will allow for a stronger interaction. Then we sort of paused our discussion at transcriptional regulators. Now, what do we mean by this? What are transcriptional regulators? We have talked about DNA protein interactions. That is our focus. But keep in mind that regulation is a context dependent term. Regulations does not always simply mean a positive interaction or a negative interaction. It simply means the ability to control, modify, manipulate the expression of a gene depending upon a number of different stimuli. What sort of stimuli? One, whether the cell has a sensing system to detect whether or not that gene needs to be expressed. Let me give you one example. Usually, if you are working in the field of biology, you will find plasmids or constructs which are sort of which are responsive to the presence of a certain drug. Commonly, you will find doxycycline or tetracycline inducible promoters. So, if they are present, or when that drug is present, only then expression of that gene is carried out, and that is one possibility. Now, the monomer of a typical transcriptional regulator is able to recognize 6 to 8 nucleotide pairs of DNA. So, what is this actually allowing this space to do? So, it is basically able this transcriptional regulator is basically able to find its motif, find its recognition sequence throughout the entire genome, and then it is able to bind. Now, another question might come here. Why is it so important for it at least to be, for that protein's binding sequence to be at least 6 to 8 base pairs of DNA? It is very similar to the concept that you use when you are designing primers for qPCR. When you're not designing primers for qPCR and instead using random hexamers. So if you knew that random hexamers are usually chosen because of the probability that the same stretch of hexameric sequence residing and non-specifically within the genome is very low. It's only one in around 4,000 bases, which is quite low. And secondly, this sequence, specifically this sequence being present, is quite less probable in the genome because not all parts of the genome are exposed at all parts of the time, at all points of time, right? So only when transcriptional activation uh, has been initiated by the cell at a certain location at a certain point of time, only then is that region of the chromatin opened up, and only then is that region of the chromatin accessible. So when a certain region of the genome is transcriptionally activated when the modifications are signaling that transcription needs to be on, only then will these regions be opened up, and only then can the transcriptional regulator actually bind. So sequence specific DNA binding proteins usually will typically have around 6 to 8 nucleotide pairs of DNA, and that is good enough. That is doing the job. Has been selected by nature over time as the most feasible sequence stretch to be present. However, if that sequence was too long or too short, A, the possibility of non-specific binding would increase, and secondly, the affinity of that binding will also make a difference. Why so? Let us assume that there's a protein um, which finds extreme which possesses extremely high affinity for a particular DNA sequence. In that case, the transcriptional control or that regulation which we just talked about would be lost. Why so? Because then it becomes such a positive interaction, such a strong affinity dependent interaction, then that switching that signal off or controlling um, the sensitivity of that signal would be lost. And in that case, the gene expression would be continuously switched on or off depending upon how that transcriptional regulator functions. Therefore, for any transcriptional regulator to be a good regulator, its binding should also be transient. It should be strong, but not so strong that it is constantly bound. Secondly, the optimal sequence or Secondly, the optimal sequence for DNA binding is not usually the exact same sequence in all places. Usually, what you'll see, for example, if you look at this sequence, which has been derived from JASPAR. So in JASPAR, you will find it's a database of protein recognition motifs for any protein of your interest. If you wish to sort of any protein which is a transcriptional regulator or a transcription factor, you will find this information from that database. So it will usually give you a pattern of this sort where you find some bases should be written much largely and some to be written, sometimes you'll find multiple bases to be present. What does this mean? It means that this is the most consensus sequence for that transcription factor. Now, what do we mean by the consensus sequence? It means that in this stretch of, I think, 7 nucleotides, these two positions, position 2 and 3, are conserved A's or conserved adenosines. However, the other sequences or other positions within this recognition sequence, they do show some flexibility. What do we mean by that flexibility? You sometimes will find T, sometimes will find C at position 1, but never G or A. Similarly, for each of these positions, you will find some consensus sequences which when present, allows the binding of the transcription factor you are concerned about. So this minimum flexibility, very similar to wobble-based pairing, what we have already discussed, allows for that flexibility or that relaxation for DNA binding by these proteins and then their control. Cis regulatory sequences, therefore, are often depicted as these logos, as I have shown here, which display the range of sequences that may be recognized by a particular transcription factor. So this depicts the cis regulatory sequence for NANOG, which is a homeodomain member transcription factor, which has a role in development of embryonic stem cells. This shows you how the shows that the protein can recognize a collection of closely related DNA sequences and gives the preferred nucleotide pair at each position. Cis regulatory sequences are read as double-stranded DNA, but only one strand typically is shown in the logo. Representation of the cis regulatory sequence as a colored box. So many transcriptional regulators, they form dimers, homodimers and heterodimers. In the example shown, 3 different DNA binding specificities are formed from 2 transcriptional regulators. What do we mean by that? So here you have your transcriptional regulator. It can form either dimer A or dimer B, or there can be a heterodimer between monomer A and monomer B. In the simplest case, the collection of non-covalent bonds that holds these above dimers or heterodimers together is so extensive that these structures form obligatorily and never fall apart. In this case, the unit of binding is the dimer or heterodimer, and the binding curve for the transcriptional regulator has a standard exponential shape. What do we mean by that? What are we trying to say? What we are trying to say is that each of these, there is a very fine balance that needs to be maintained by the cell, because again, we are calling it that the cell is dynamic. All these interactions are transient, and it is necessary for these interactions to be transient for us to not have any bias in our studies. So non-covalent bonds are the major drivers in this case. Why? Because any covalent bond, A, would be energetically very expensive, B, would be a huge waste of energy if not allowed to sustain for an extended period of time. So any interaction which is transient or responsive to stimulus or is subject to change depending on what stimulus is received by the cell, any such interaction needs to be facilitated, needs to be controlled by non-covalent bonds. And in many cases, these dimers and heterodimers are held quite weakly. They can predominantly exist as monomers in solution, but they can form dimers based on what is the condition. And what you will see is that the binding curve for the transcriptional regulator is usually of a standard exponential shape. And that exponential shape explains something which we'll come to very soon. In many cases, which we were discussing, dimers and heterodimers, if held very weakly, they can predominantly exist as monomers in solution, but on the DNA sequence, they might form dimers. So again, keep your eyes here. You will see that there's a possibility that there can be very rapid exchange or interchange of these um, different states. So why are we so worried about this? If you look here, non-cooperative binding by a stable heterodimer, that's what the legend says. It means that in solution, the two monomers are able to stably form this heterodimer, and the balance or the equation or the equilibrium is maintained between a DNA bound and free solution heterodimer, and therefore in that case the curve which is formed, the binding curve which is formed, is exponential in nature. However, when there is cooperative binding, then over a range of concentrations of the transcriptional regulator, binding is more often all or none phenomenon than for non-cooperative binding. What does it mean? It means that at most protein concentrations, the cis regulatory sequence is either nearly empty or nearly fully occupied, and it is rarely somewhere in between. Here, the curve is different. It differs from that of A because the fraction of protein in a form which is competent to bind to DNA, which is the heterodimer, will increase with increasing protein concentration. When your protein concentration increases, then in solution, the possibility of that heterodimer forming will increase, and therefore the occupancy of DNA or the fraction of DNA which is bound will increase as well with increasing protein concentration, and you will get a curve which is similar to this, which shows you sort of a sigmoidal curve, which shows you cooperative binding. So in most cases for transcriptional heterodimer formation, you would find some sort of a cooperativity to persist. The cooperative binding of transcriptional regulators to DNA occurs because these monomers have only a weak affinity for each other. The formation of stable heterodimers is not the default option. However, there is a second, rather sort of indirect mechanism for cooperative binding as well, one that arises from the nucleosomal structure of eukaryotic chromosomes. So, what is that? In general, transcriptional regulators, they bind to DNA DNA that is in the nucleosomal structure. They bind to such DNA with very low affinity compared to the affinity they have when DNA is naked, when DNA is open and accessible. There are two reasons for this difference. First, the surface of the cis-regulatory sequence, which is recognized by the transcriptional regulator, might be facing inward. So the transcription factor is not able to find its motif, and therefore it's not able to bind, because it is because that motif is sort of uh, it has bent in a way such that it is now facing the histone core and is no longer accessible. That's one possibility. Second, even if the face of the cis sequence is exposed on the outside, many regulators subtly alter the DNA conformation when they bind. And these changes just give me a minute. Yeah, okay. Sorry for the disturbance. So, yeah, like we were saying, it's possible that sometimes your DNA is not accessible because, or your transcription factors are, or transcriptional regulators are not able to bind um, their cognate DNA sequence because the DNA is facing away from them and it is instead facing the histone core. The second possibility that even if it is exposed, some transcriptional regulators need to sort of sit on the DNA, which means that they need to completely encompass the DNA and they need to change the conformation of DNA when they bind. And therefore, they need the complete naked DNA for that purpose. And that would be opposed or would be impeded by the occupancy of the DNA inside a nucleosome or when it is wrapped around the histone core. So mostly that happens when transcription factors induce a sort of bend or a kink in the DNA when they bind. Then, what is the solution? Because this would be the case for most of the transcription factors, but despite all of this, even in a partially unwound genome, transcription does proceed. So, what exactly happens? Something very important and something we have extensively discussed that occurs is nucleosomal remodeling. What is nucleosomal remodeling? It alters the structure of the nucleosome or chromatin remodeling. Otherwise, it is also known as chromatin remodeling, whereby the chromatin is opened up, closed, sort of separated to open up certain regions. All of these modifications are possible. And this allows transcriptional regulators some access to the DNA. Even without remodeling, however, transcriptional regulators can still gain limited access to DNA in a nucleosome. The DNA at the end of a nucleosome breathes. This is nucleosomal breathing. So this is your histone core around which your cis sequence is bound, and a very small region is exposed because this binding is sort of dynamic. There is some breathing which occurs. This open form occurs about 1/20th of the time, but in that limited duration, this transient exposure of the DNA can allow some regulators to bind. But of course, this breathing happens at a much lower rate in the middle of the nucleosome. Therefore, the position of exit and entry of the nucleosome, they are much easily accessible. So if such regulatory regions are present, then it's one possibility. Second, if a regulatory protein enters the DNA of a nucleosome and prevents the DNA from tightly rewrapping around the nucleosome core, then it will increase the affinity of a second transcriptional regulator for a nearby cis regulatory sequence. What do we mean by that? If there is a regulatory protein, for example, uh, let's go to the figure here. So compared to its affinity for naked DNA, a typical transcriptional regulator can bind with 20 times lower affinity if the cis sequence is located near the end of a nucleosome because of the breathing possibility. Now, what is the other possibility? If a regulatory protein is such that it enters the DNA of a nucleosome and prevents its rewrapping and it keeps that region open, then it will increase the affinity of a second transcriptional regulator for a nearby cis regulatory sequence. Yeah. If the two transcriptional regulators also interact with each other, the cooperative effect is even greater. In some cases, the combined action of the regulatory proteins can eventually displace the histone core of the nucleosome altogether. So in that case, what happens? There are two transcriptional factors which are then interacting with each other. The cooperativity increases. So if more and more amount of transcription factor A shown in green increases, more DNA would be exposed, more amount of transcription factor B will be able to bind, more amount of transcription will occur in this region, and that will facilitate increased binding of both of these proteins. So in this case, cooperativity would also be enhanced when the combined action of both of the regulatory proteins can be seen together. So taken together, that is what I wanted to discuss for today, where we have introduced DNA protein interactions. In the next class, I will be shifting to techniques that are used to study DNA protein interactions. We'll begin with the lab driven techniques, and later on we'll in the next class we'll move on to high throughput techniques for detecting such strategies, and then we'll shift to analysis of such techniques as well. So that is where I would like to end today's class. Thank you so much for joining. I'll see you again next Tuesday. Good night.